This is the key assessment one for the Upper 6 core concepts multiple choice, which produced much hilarious thinking. So here we go. Here's just some help with this. Right, here's a circle. There's pi. Let's have a look. What's the area? So we're area. Okay, area is pi r squared. So some students could not remember what the area of this circle is. And then all you need to go is what's the radius? What's the radius? And there's the stage. That's the eyepiece. So there's the stage. Division's 0.1, so that's 0.1, that's 0.1. And then if we move this down a bit, so that would be 0.1, 0.2 and a half, 0.25. In micrometers, 250. So the diameter is 250. And now you can probably do it. If the diameter is 250, then you're not going to use 250 in your calculation, are you? Structures found in both animal and plant cells. Right, well, you need to sort of think, right, there's one that's definitely, and it certainly seems you're doing the cell division topic at the moment. One of these is definitely not found in one or other. Okay, so if you looked at your revision notes, you'd find in the table not in certain type of cells. Some problems with the molecular structure of starch. So you need starch, amylose and amylopectin, and you need to know, coiled, branched, and stored energy. So structure. So all pretty similar. All pretty similar there. So he's most suited. Oh yeah, okay. You're on your own with that one. Question 4. Diagram represents the ring form of beta glucose. Well, you need to sort of go, what do I know? So I know alpha glucose. So alpha glucose is, if we draw it like, so they're all the sort of same shape. Alpha is down, down, up, down. So that's alpha. And now you're going to go, right, which one changes in beta? And then what's it look like? How many fatty acid residues are normally present in a phospholipid? So here's a phospholipid, glycerol part, phosphate, and then we've got, that's what it's going to look like. No excuses there. Then frozen lake water, mineral nutrients are brought to the surface. Well, why does anything move to the surface? It's got to be something to do with density, hasn't it? And then these are all true, which is a bit of a problem. But you can't have it for they're all true, and they kind of are. My sympathy, because I would think, well, that's relevant. That's definitely relevant. And then, well, but it wants things moving to the surface. So it's got to be something to do with density, and then mineral nutrients. So they've got to dissolve. And 2, yeah, true, but maybe not so important. So you think, well, I want to say 1, 3 and 4, but I can't. So I've got to go for the one that's most like that, with the most important parts. Question 7. It starts from glycogen. Well, it's one of these components is slightly different to the general. Glycogen just really has one kind of structure, whereas starch has 2, and the difference is in one of them. So fairly technical, but you should know that. You should know that. And there's some surprise questions from component 3. And flow of current through the atria. I think you were fine there. and then recovery of the ventricle walls. Well, the clue is the recovery of the ventricle walls will happen quite late on in this cycle. And what is systolic? So we should be fine. It's systolic. Where's it going? Where do you measure this? So when they test your blood pressure, where do they put the sphygmomanometer? Where do they stick that? Do they stick it in your ventricles, or they wrap it around your arm? Or there's a clue that should help you decide which part. And question 10. I could come up with a couple of answers here, and then it's the important thing is you can get rid of a couple of wrong answers, and then you're left with a couple that you think, oh, okay, and then going for one of those.